Hello everyone, it's me Shanty and you're watching Biology Nowadays. In this video, let's discuss about seed dormancy with a focus on the two types of seed dormancy, namely seed coat dormancy and embryo dormancy. We will also discuss about the natural and artificial processes for removal of seed dormancy. Normally, viable seeds, don't germinate as soon as they are formed. They will be in a state of suspended growth. The suspension of growth before germination could be sometimes due to unfavorable external environmental factors. For example, if the seed didn't receive the right temperature, light, or water, it will not germinate. Sometimes the suspension of growth of the seed may be due to internal control within the seed, like hard seed coat, immature embryo, presence of germination inhibitors, etc. The external control is called the exogenous control, while the internal control is called the endogenous control. If the suspension of growth of a seed is due to the exogenous control, it is called quiescence. On the other hand, if the suspension of the growth is due to the endogenous control, it is called dormancy. In this video, we will be focusing on seed dormancy. Now, for a better understanding of seed dormancy, consider a situation. Suppose that I collected some rice seeds and I sowed 2 rice seeds in the soil. I provided all the favorable environmental conditions for germination, like the right temperature, light, and water to both these seeds. After a few days, I observed that only one seed had germinated. The other seed didn't. That is, even though I provided all favorable environmental conditions, this seed couldn't germinate. So it is clear that the reason could be the endogenous control within the seed itself. Now we can say that this seed is undergoing a period of dormancy, or let's call this a dormant seed. A dormant seed is actually a viable seed, which may stay dormant for years or even centuries. It will germinate once the reason for dormancy is removed either naturally or artificially. We will discuss about the natural and artificial processes for removal of seed dormancy later in this video. By the way, a dormant seed is different from a non-viable or dead seed because a non-viable seed will not germinate in any case. So, seed dormancy can be defined as a physical or physiological condition of a viable seed which prevents its germination even in the presence of favorable environmental conditions. So, now let's discuss about the reasons for seed dormancy. Seed dormancy may be either due to seed coat dormancy or embryo dormancy. Let's see one by one in detail. First, seed coat dormancy. It may be due to a hard seed coat that is impermeable to water. This condition is very common in the seeds of many legumes, water lotus, morning glory, etc. Next reason for seed coat dormancy could be a seed coat that is impermeable to gases. This is found among seeds of many grasses and cocklebur. As you know, when seeds take up water, the metabolic processes initiate with an increase in the respiration rate. As a result, carbon dioxide inside the seed increases. But since the seed coat is impermeable to gases, oxygen cannot enter inside the seed to facilitate respiration. Thus, those seeds fail to germinate. Another reason for seed coat dormancy could be the mechanical resistance of the seed coat to embryo growth. This is found in the seeds of certain weeds like pigweed, shepherd's purse, water plantain, etc. The seed coats of these plants provide mechanical resistance to expansion and growth of embryo, preventing the embryo from further development. Seed coat dormancy can be removed by weakening or breaking the seed coat. Any process of breaking, scratching, or mechanically altering the seed coat to make it permeable to water and gases is known as scarification. Scarification can be achieved by natural or artificial processes. The natural processes include microbial action, mechanical aberrations during seed dispersal, passing of the seeds through the digestive tract of birds, animals, etc. When birds and animals consume the seeds, it is the hydrochloric acid and certain enzymes in their digestive tract that soften these hard seed coats. On the other hand, the artificial processes of scarification include either rubbing the seed with a sandpaper, nicking with a knife, pricking it with a pin, or cracking gently with a hammer to weaken the seed coat. Scarification of seeds can also be done by soaking them in hot water or even in concentrated sulfuric acid. Sometimes seeds are subjected to hydraulic pressure to break dormancy. Now let's discuss about embryo dormancy. It may be due to immature or underdeveloped state of the embryo at the time when seeds are shed from a plant. For example, in plants like ginkgo biloba, European ash, and many orchids, the embryo is unorganized when the seeds are shed. These seeds will germinate only after the embryo attains full development. Another reason for embryo dormancy could be the presence of germination inhibitors in the embryo, endosperm, or other tissues of seed or fruit. Some examples of germination inhibitors are abscisic acid, phenolic acids, ferulic acid, coumarin, para-ascorbic acid, etc. Among all these, abscisic acid is one of the most commonly detected germination inhibitors. The effect of inhibitory substances can be removed by subjecting the seeds to chilling treatment or by application of gibberellic acid and nitrates. Even though seed coat dormancy results in a delay of germination, there are some advantages too. It serves as an adaptation to ensure that the seed germination occurs only under favorable environmental conditions. It enables the embryo to overcome the unfavorable period of the year like winter. It enables the storage of cereals, millets, pulses, etc. to be used later for food or for sowing in the next season. If you find this video helpful, hit the like button and let us know in the comment box below. Also, don't forget to subscribe this channel for more biology videos.